So for part B here, there's no experiment, no experimental video. A couple reasons for that. There's really not anything to see, honestly. I mean, a lot's going on chemically, but you start with clear and colorless solutions and you end with clear and colorless solutions. So again, not much to see. If you did this experiment in lab, really the only observation that we would see for this class would be and we wouldn't see it, we would smell it. We'd smell the old odor of the ester. So esters they're fairly sweet smelling compounds. Each of these reactions produce a particular ester. For example, and I'm going to actually fill out this row here for you. If you were in the lab and you did this experiment, what you would smell would be wintergreen. Now, in addition to recording the odor of the ester, well, the other two parts would be writing in the reactants and then drawing and naming the product. So let's do that for part A. So again, I'm going to do part A for you. And again, this first column, these are the reactants. Again, just like all these labs, you really need to watch the lecture videos before doing the lab. That's what I would recommend. And what you see there is an alcohol reacting with a carboxylic acid then yields this ester. Now methanol has a fairly small structure. It's the smallest alcohol. And then salicylic acid has this structure. So it's an aromatic compound, has a carboxylic acid at one position. And then at the second position over, what you can call the ortho position, then you have a hydroxy group. So really you have a phenol functional group within salicylic acid. Now the reactivity here is occurring between the alcohol and this carboxylic acid. So this is a connection that's going to be made. Let's take a look at that. So I'm going to start writing this just because that's typically how it's presented to students. The phenol portion is unchanged. The ring structure is unchanged. It's just this is changed. Remember what happens. The R group on an ester basically replaces the proton on the carboxylic acid. Now, I've said this a few times here and there in this class. We don't look at the mechanisms for these reactions. And so what I'm describing here is not specifically how the molecules collide and change and go through transition states, etc. I'm just showing you what it looks like going from reactant side to product side. Okay, so again, there's an oxygen here, and then just this R group, the methyl group. Now, usually, I don't know if it's just out of habit for me, but I always add water. These are condensation reactions. Water is always kicked off in a esterification reaction. You don't need to write that down if you don't want to. Because again, we're just looking for the structural formula, which is this one, and then the name of this ester. And then when we name esters, remember the basic form of it. It's a two-part name. First one is the alkyl group from the alcohol. So in this case, it's going to be methyl. Let's just go ahead and ink that in. And then the second part of the name is you take salicylic acid and then change the IC to an ATE. So basically you're going from the carboxylic acid name to the carboxylate name. And remember what I said. You just change that ic to an 8. So this will be methyl salicylate. And experiment A is complete. So what I want you to do is do the same thing for all the remaining, four reactions that are illustrated here. And then what you can do is that once you've identified what ester is formed, just Google that ester and you'll see what odor it's responsible for. So for this column, the observation, just find it online. And again, there's no experimental part here. So there's no video. But now for part C, we do have a video for the saponification. And of course, we'll see that in just a minute. What happens in a saponification reaction? Well, you take an ester. I'm using a little prime here on this R group just to represent that this is what's associated with the alcohol part of the ester. Now, saponification is also known as base hydrolysis. And so what you see here is that some strong base, it's usually sodium hydroxide or potassium hydroxide, and then you would heat it up. Now, actually in the video, you're not going to see the heating part because it's just a clear and colorless solution going to another clear and colorless solution. The video that you'll see here in a minute is what happens at the tail end of the reaction. And I'll get to that in a minute. Now this base, what it does is it breaks. You can imagine it sort of, well, in some ways you can say it breaks right there. You reform the alcohol. I'll just put it on this side, so it would be R prime OH. And then since we're in a basic environment, when it's not the carboxylic acid that's formed, it's actually the carboxylate compound that forms, or the carboxylate ion that forms. That base prevents this group from being reprotonated. So what happens is that this thing is oftentimes soluble. Like I said, we're starting with a clear and colorless solution, and we wind up with a clear and colorless solution. That's because the product that's formed here is an ion, and it's just soluble in that solution. So again, you don't see any changes. That's why I'm not doing showing you the heating step for this part. So in part A, please draw the structural formula. It's going to be for methyl salicylate. It's actually the same one that we saw in part A, experiment 1 of part B. So its appearance, well, I guess I've already said it. It is clear and colorless solution. And I'll just tell you, the odor originally is wintergreen. Now, once the saponification occurs, the changes, well, the odor, there's no odor. So this change in the odor of the sample suggests a chemical reaction has taken place. But in terms of the appearance, you'll see it in the video. Again, this is the beginning of the video, and I'll make a note in the video when you start watching it. But it's a clear and colorless solution. Again, you can't smell an odor on video, so maybe that's another reason why I'm just not showing this step. Now, this part 4 here, what you're going to do is you're going to take methyl salicylate and then run it through this reaction. So methyl salicylate will here, and then you're going to break it into its carboxylate salt and then the alcohol that's coming off of that compound. Now, this is where the video will start. Now, this observation is when HCl is added. So you can record the changes. And then based on that change, then you should have some idea of what the condensed structural formula is for the compound that's formed when HCl is added back to the sample. And I just want to give you a quick little heads up. Go back and take a look at part A, number 6. This is what's happening. Carboxylate salt, there's HCl being added, and then that reforms. And remember, when it goes from a carboxylate to a protonated or carboxylic acid, then this has a lower solubility. Just based on that description, you can actually anticipate what's going to happen when the acid is added here. But again, it's just this step in parts for the video that you're about to see, so it's pretty fast. And that's it for Lab 25. Thanks.